Okay. Hey, so hi, my name is Dhiya Sudini, and I developed CRISPR Guardian. So CRISPR is a gene editing technology used to repair DNA sequences. It identifies mutations and mismatches early on, but sometimes it can have a few errors. So now CRISPR is used to advance biotechnology, engineering, and help in various healthcare causes. So globally, millions of people have genetic disorders. This is due to a lack of knowledge of how these disorders come about. So scientists wanted to create a technology to identify where these errors are at a microscopic level by actually opening the gene and seeing where the mismatch occurs. So in the real world, CRISPR has been used to improve a lot of lives. It's been used to treat genetic disorders such as sickle cell disease and B thalassemia, and has been reported to improve the quality of life and reduce painful symptoms. It's also been useful in improving cancer research. It's been used to aid immune cells to target cancer cells early on, and has also been used to improve detection of pathogens to prevent progress of infectious diseases. So CRISPR Guardian is an AI software tool. It includes 12 features, one of them being ranking DNA and RNA sequences from safest to riskiest. Through this software, users will be able to identify genetic disorders early on by seeing the percentage of mismatch errors within sequences. And this is also just the basis of CRISPR. Through more development and programming, we'll be able to um, see more sequences and really understand where the genetic disorders are coming from. Okay, so here's a demonstration, and I can. Okay, so this is the landing page. This landing page includes 12 of the features here and a rundown of how the model works. And then here you can open the software. So this first line just gives you basic information of how the software works. Here's a chatbot. I can type in any question about CRISPR and DNA. For example, if I type in, what is CRISPR? It'll give me a 5 bullet point list of information of what it is. The single predictor function, it compares a guide RNA sequence with a target DNA strand. So for example, if I change the sequence, it gives me a classification output. So in this case, it's a very low mismatch count. Therefore, it's only 4.95% of error. And this image shows exactly where this error occurs. In this case, it's the last letter. The bulk comparator compares an RNA strand against target candidates. And 3 is just a minimum. It can compare various different ones. For example, if I type in this, 4, it'll rank from number 1, which is the safest, having the lowest score, to the riskiest, which is the highest percentage of a risk score. The genome track compares, or it has the x-axis, which is the position within the chromosome, and then the y-axis shows the off-target risk score. And you can see that like the higher peaks show a higher risk score, while the lower peaks are lower risk score, and it changes over time. And you can also zoom in to get like a greater detail. So the tensor style matrix, you can type in a sequence like, let's say this, for example, and you can change it around a little bit, and you can see the colors changing. So a high color, or a yellow, between this case a T and a T, shows that this is a very high mismatch error, while a dark purple shows a low mismatch error. And you can reference it by looking at this key. The genome heat map is connected to the genome track. So the areas of red is a very low probability, while the areas of yellow are high. And you can zoom in and see. And you can compare this to the actual genome track. So the red areas would be over here and the yellow would be over here. Okay, the demo analytics. So as users use this app throughout time, the probability score of a given sequence can change as they identify the errors and fix problems. So as you can see, like over here, the error starts to increase, but then there's some success rate over here. So the safe CRISPR score, it takes a composite score of 100, and then it subtracts the amount of errors, mismatches, and GC content errors, and it gives a cumulative score. So if I type in something like this, it'll give me an output score. So in this case, it's 51.7, which is basically in the middle. And it gives you also the risk information and the amount of mismatches and the error over here, the amount of G's and C's. So the risk factors, if you explain the risk factors, it'll give you information about the features like the mismatch count, seed region mismatches within, and the amount of G's and C's in the nucleotides. And it gives you an explanation of like why this happens. For the recommended alternative guide RNA sequence, I type in a guide RNA and it compares it against the DNA. And if I ask it to find a safer alternative, it'll compare possible candidates and it'll give you the safest percentage. In this case, 75.18 is the lowest, so it'll give me that as an alternative sequence that can replace this given sequence. The SHAP explanation plot, if you ask it to generate a chart, in this case, it shows that the GC content is the most likely or the greatest contribution to the risk compared to the other two. But this is not always the case. So if I change this up, it can be that it's seed mismatches in this RNA sequence is the main contributor to the risk. The multi-model comparison, this is more of a reliability. So this tests how this risk score compares to different machine learning models. So for random forest, it's 71.8, but for the other two, it's way higher. But then again, they're all high risk. So it just references the mismatch along different sources. The model performance evaluates this whole program and it shows kind of like a reflection on how well it did. In this case, it classified 5 out of the 6 correct. And it shows exactly which one I did wrong. And this is just to show that like it is still an educational simulation and it's not like, again, used completely for medical purposes just yet. So it's just like to tell the user that this is just a simulation. And yeah, that's it. Thank you.
I mean, it has a lot of features and a lot of visualizations. So, did you do the visualization? Did you came up with this visualization yourself, or did you took inspiration from somewhere?
Like as in the graphs?
Yeah,
Yeah, so our team kind of, like our group kind of thought about features we could do. And based on the features, some of them needed graphs. So like, for example, like the genome track, like the only way to do this to show the data would be a visualization because it's just easier. So I wanted to include some images. So instead of just information to like kind of just mix it up a little bit. So yeah, it was kind of like a group idea, but I was the one that kind of like tried to figure out how to include it in my dashboard.